Hi Manju, I'm just going to make this brief video about HFI to try to explain it a bit more. So when you eat fructose, um, well let's say you eat a piece of fruit, right? This is going to have fructose and glucose in it, and the glucose is used by all the cells in the body, and so it can go in the bloodstream and get absorbed by any cell in the body. Fructose, on the other hand, is only metabolized in the liver, the kidney, and the small intestine. This is giving me a little bit of trouble here. Okay, but mostly in the liver. That's where most of this happens. Okay, and so all fructose is going to go to the liver. So let's take a brief look here at what happens after you eat that piece of fruit. Okay, so this is the liver enzymes after you have a meal or after you have something that has glucose and fructose. So you can see right here that the glucose comes in to the liver, and the glucose is phosphorylated to give this glucose 6-phosphate, and that goes to make glycogen. Okay, so like to store up some glucose. And the fructose comes in, and the fructose is phosphorylated by fructokinase to give fructose 1-phosphate, or F1P. And this is the substrate for aldolase B. Okay, and aldolase B, I was just going to circle this. Here we go. Aldolase B turns the fructose 1-phosphate into DHAP and glyceraldehyde, which can then go into glycolysis. Okay, so this is glycolysis. Okay, that's what makes the pyruvate here, which can then lead to fatty acid synthesis, or if the body needs energy at the moment, then it will go into ATP synthesis in the electron transport chain. Okay. Now the other thing that can happen though with this DHAP and glyceraldehyde is it can actually go this direction, okay, back to the fructose 6-phosphate and glucose 6-phosphate. These are also intermediates in glycolysis. And when we go backwards through glycolysis, you may remember that this is known as gluconeogenesis. Whoa, that's an S. Gluconeogenesis, which basically means that you're making glucose. Okay? And you may ask, why would we do this? Well, remember, the liver is an important organ for lots of reasons, and one of the reasons is that it makes this glycogen. It stores up glucose. When you've had a meal and there's lots of glucose in the blood, it will store up excess glucose. And then when the body, the rest of the cells in the body have kind of burned through their glucose and they need more, then the liver will break down this glycogen by sending it back to glucose 6-phosphate, and then this phosphatase will take the phosphate off and give glucose, and that glucose can be transported back out into the blood, okay, to be carried around to, oh, that's really unclear. Let me just write that again. Yeah, so out into the bloodstream, and carried to the cells that need it. Okay, and so when fructose 1-phosphate is broken down, it contributes to both of these processes, both glycolysis for making ATP or fatty acids, as well as gluconeogenesis for generating glucose that can be stored in the liver as glycogen or sent out into the blood to help when blood sugar is low. Okay, so let's now look at if there's, after it's been a while after the meal, then this is what happens, okay, is, again, we kind of talked about this a little bit, but the glycogen, well, where am I? Right here, glycogen is going to be breaking down to the glucose 1 and then glucose 6-phosphate, and then the phosphate will be taken off so that glucose can go out into the bloodstream. Okay, so that's normally what happens. And partly because this glucokinase, it gets, its activity is kind of blocked, okay, during this time. Because if it wasn't blocked, then it would keep phosphorylating glucose, and glucose would not be able to leave the liver, because once it has a phosphate on it, it stays put inside of a cell. And so when the liver is trying to break down glycogen and send glucose out to the body, it blocks this glucokinase here. Okay, so now what's going to happen if aldolase B is no longer working, if we knock this out? Well, we know fructose 1-phosphate is going to build up. And there's a couple of problems with that. One is that in order for this fructokinase to put the phosphate on fructose, it actually uses an ATP. Okay. So we get ADP, and the phosphate is here on fructose 1-phosphate. And normally that's not a big deal because it gets broken down and goes through glycolysis and generates more ATP. But if we get stuck here and fructose 1-phosphate starts to build up, this phosphate is stuck, okay? Which means, but as long as there's fructose around, this fructokinase will keep phosphorylating the fructose. So we're using up ATP, we're using up phosphate because the phosphate's not coming off, it's not going back into the cycle. And what that does is it depletes phosphate stores, okay, inside the liver cell. So the phosphate pools in liver go down, okay? And that's a problem. And the reason it's a problem is because we need the phosphate to generate ATP, right? So if you remember in the electron transport chain, in the last step, we have this ATP synthase that lets these hydrogen ions come through in a controlled way. And when it does that, it takes ADP plus phosphate, okay, that stands for inorganic phosphate, that's how it's often written, to make ATP. So if there's not a lot of phosphate around, that's going to hinder ATP production, okay? Which means that the liver cell is starting to run out of energy, all right? Because we're blocking this here, right? Now the other problem is normally, if aldolase B was functioning, then this DHEP and glyceraldehyde would be used for gluconeogenesis. Remember, we've got gluconeogenesis going on here. Genesis. And because these are no longer being made by the aldolase B, this gluconeogenesis is also getting blocked. And that is a problem because then the liver has a harder time making the glucose to send out into the blood. And to make matters worse, fructose 1-phosphate actually acts as a signal that gets rid of this blocking of glucokinase. And so even when glucose does get made in the liver, the glucokinase puts a phosphate back on it so it can't go out into the bloodstream. So it's kind of a two-edged sword here with the fructose 1-phosphate. They can't generate ATP in the liver cell because this part is blocked and because the phosphate's getting used up here. Gluconeogenesis is slowed down, so it's harder for the liver cell to make glucose. And even when they do make glucose, before it can ship it out to the blood, this glucokinase, which has been activated by the fructose 1-phosphate, puts the phosphate back on glucose and it can't leave. So so what we end up with here is gluconeogenesis slows down, or you could even maybe say stops, but it doesn't fully stop, it mostly just slows down. ATP production is slowed down. I'm going to use arrows. Okay, so let's do this. To make this clearer, hopefully. All right, so we've got gluconeogenesis goes down. ATP synthesis, because of phosphate pools, phosphate stores pools, both of these go down. And export of glucose to the blood also goes down. Okay, the only thing going up here is fructose 1-phosphate. Okay, concentrations. These brackets mean concentrations. So concentrations of fructose 1-phosphate. And this is a real problem, right? Because if the liver cannot help to regulate blood sugar, then the rest of the body starts to feel hypoglycemic, meaning not enough glucose in the blood. And that's because the glucose is getting tied up here, and we're having a hard time with gluconeogenesis, having a hard time exporting it, all because of this fructose 1-phosphate building up, which is a result of aldolase B being knocked out. Okay, and it's these symptoms of hypoglycemia and also liver problems. So this also leads to some liver problems because the liver cells can't make ATP very well, right? And so they end up having low phosphate, low energy, and eventually the liver cells will die. And so we have these liver problems and these hypoglycemic symptoms that are associated with HFI. Okay. So hopefully this helps to clear up how aldolase B being deficient or being kind of knocked out, how that leads to HFI. And it's all through this fructose 1-phosphate buildup. Again, through the phosphate stores, blocking ATP, blocking gluconeogenesis, blocking glucose export to the blood. All these different things lead to these symptoms of HFI. I hope this is helpful.